Hi guys, welcome to the liver and gallbladder histology. Right, so the liver is the largest gland in the body. Um, it has 5%. It takes 5% of total body weight, and it is involved in up to 200 functions, which is very important. One of the functions are synthesis and secretion of bile, which is used for emulsification of lipids right in the duodenum. It also stores glycogen and become a lipid reserve. Remember the action of insulin and glucagon affecting the glucose concentration in the blood. Metabolism of the carbohydrates, fats, and proteins, as well as storage of ferritin, which is iron, detoxification, synthesis of plasma proteins, albumin, clotting proteins, very important, right? And they also phagocytose damaged red blood cell, producing the bile from bilirubin. And in utero, liver it's a hematopoietic organ, which means it produces red blood cells. Now, we remember from embryology, we said that the liver it has dual origin. The parenchyma of the liver, which are the hepatocytes, which we're going to talk about, they come from the endoderm, while the connective tissue, the Kupffer cells, as well as the hematopoietic stem cells, they come from the septum transversum, which is the splanchnic mesoderm that will be surrounding the heart. So let's take a look at the stroma, which is the supporting unit of the liver. First of all, a liver is an organ, so there is a capsule, a thin capsule, which is called the Gleason's capsule. So this Gleason's capsule, it will send septors dividing the gland into lobules as always. The connective tissue sheath surrounds the larger vessels from the hilum up to the portal spaces, right? This connective tissue sheath. You also have epithelial cells and sinusoids making up the liver parenchyma, right? And these are supported by reticular fibers, which is type 3 collagen. So we have a capsule that sends septa into the liver, dividing it into lobules, and the connective tissue, right, that will surround larger vessels, right? And we also have epithelial cells and sinusoids making up the liver parenchyma, right? And the parenchyma is going to be mainly supported by delicate reticular connective tissue, which you're going to see very soon. So as you can see, this becomes your Gleason's capsule. That's a thin capsule of the liver which dense irregular connective tissue. You can actually see it is sending trabecules or just septors into the liver parenchyma, dividing it into lobules. Right. So you can actually see these stroma, the stroma of reticular connective tissue, right? Um, Supporting your hepatic cells. So these are your hepatic cells that are being supported by this reticular connective tissue, right? You can actually see the reticular fibers, they are actually abundant here, right? So these are your reticular fibers. So now let's go to another video, right? So a principal cell called the hepatocyte forms the parenchyma of the liver. Hepatocytes are arranged into plates and cords that are one cell layer thick. So they are arranged into plates that are one cell layer thick, right? So the cords and the plates of the hepatocytes are separated by a large discontinuous capillaries called liver sinusoids. Remember, sinusoids are discontinuous capillaries. What does discontinuous capillary mean? It means that they have a discontinuous basal lamina and fenestrated endothelial cells, right? And there's going to be a small space between the sinusoids and the hepatocytes. That is called the space of Dis, or the subendothelial space, right? So this space is very important because there are cells that are going to be found within the space, right? And substances that are going to be found within the space that you have to know, right? So the basolateral surfaces of the hepatocyte is lined by the space, lining the space of this. You have microvilli. So one thing that's found in the place of this, microvilli. Two, Kupffer cells. So Kupffer cells, these are macrophages, right? You also have Ito cells. Ito cells, these are fat-storing fibroblasts that take part in storage of fat or lipids, vitamins, right? And also they take part in the synthesis of vitamins. Synthesis of vitamins, right? So these are the following are found in the perisinusoidal space, which is the space of this. One, blood plasma, not blood plasma. So remember, these sinusoids, they have a discontinuous basal lamina, so you have the blood plasma entering the space of this. You also have those Kupffer cells, which are macrophages, the Ito cells, right, and the microvilli from hepatocytes. You understand now? Good. Now, cords and plates of the liver cells are radially orientated towards a central vein. So there's going to be a central vein that is going to receive all the blood from the sinusoids, right? And this central vein will join and form a hepatic vein. The liver sinusoids drain into the central vein. True. The liver sinusoids receive blood from the portal vein that will be rich with the nutrients, and blood from the hepatic artery, which will be rich with in oxygen. And it is this blood that will move towards the central vein. Right. So this is a good schematic diagram of what we're talking about. So you can actually see that these are plates or cords of hepatocytes. You can actually see. And this plate is separated by this space, right? What is found in this space? In this space, we have what you call the liver sinusoids. And these liver sinusoids are made of hepatic artery and portal vein, and they'll be draining blood into the central vein. So this whole thing, the hexagonal thing, is going to be called your hepatic lobule with a central vein on the center. So blood is going to move from this portal triad towards the central vein, right? So if you are to look at this portion, you can actually see these are the Kupffer cells, another Kupffer cell over here, another Kupffer cell over here, which are macrophages, right? And between the, the sinusoid and the hepatocytes, there is a space right here that is called the perisinusoidal space, out of the space of this, where we find the microvilli, Ito cells, Kupffer cells, as well as blood plasma. Good. So this is a histological structure of a section of a liver. So you actually see this is a liver lobule. This is another liver lobule. This is another liver lobule. Right? This is your central vein, central vein, central vein right here. This is a prototriad, 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 prototriad. So the central vein it receives tributaries from the liver sinusoid. So all these gaps you see, the whitish gap, these are your sinusoidal spaces. While these eosinophilic cords, they are gonna be your hepatocytes. Right, you can actually see here, this is your central vein. So I want to highlight this is a cord right here. Right, this is a cord, and between the cord you're going to find these spaces. Right, this is your sinusoid, another sinusoid over here, another sinusoid over here, another sinusoid. So you just cord and sinusoids, cord and sinusoids, cord and sinusoid. You understand? And the cord is one cell layer thick. Right, and take note of this. The hepatocytes, they can also be binucleated. You can actually see here, binucleated. It is usual, right? It is very usual to find a binucleated hepatocytes. They are binucleated. They can be polynucleated, right? It is very important. Another good diagram, you can actually see this is your central vein. These are your sinusoids, 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 sinusoids with blood, sinusoids, and these are the sheets of the plates of the hepatocytes. You can actually see them. Another diagram again, right? So within the spaces, the cells that we see within the spaces like this is your Kupffer cell. This is another Kupffer cell. This is another Kupffer cell, right? They will be found within these sinusoidal spaces. This is another Kupffer cell, right? And then this is a hepatocyte. This is a hepatocyte, which is sort of like polyhedral, but a high eosinophilic cytoplasm. Another here. You can actually appreciate here, this is a simple squamous, right? This is now an endothelial lining of the central vein, which is a simple squamous, right? Epithelium. You can actually see a flat cell right here. Now, so let us take a look at what we call the liver lobule. The liver lobule. So the liver lobule, that's the hepatic lobule, is the smallest structural unit of liver, and it is a hexagonal in shape. Hexagonal in shape, right? You can actually see this is a hexagonal in shape with 3 to 6 portal triad at its edge. There's a portal triad here, portal triad here, portal triad here, portal triad, and at the center you're going to have your central vein. So it consists of a polygonal mass of cords and plates of hepatocytes radially arranged surrounding a central vein. Right? So at the periphery, you can have about 3 to 6 portal canals at the periphery, right? And all of these portal canals will be draining into a central vein. Right. Let's talk about the portal triad. It can be called a portal triad, portal space, portal tract. Yeah, there are so many names, but it consists of a branch of the hepatic artery, a branch of the portal vein, and a branch of the bile duct. So these three makes the hepatic portal vein. It makes the portal triad, sorry. A branch of the portal vein, a branch of the portal artery, and the bile duct. But along these structures, you can also find nerves, lymphatics embedded within a connective tissue. Right, this is a beautiful diagram here. So a hepatic lobule, it is hexagonal in shape with the 6 portal space, portal canals, portal triad at its edge, and a central vein. So you're going to have a portal vein, a bile duct, and the hepatic artery forming this portal canal. So take note of this. A hepatic lobule, right? It emphasizes the endocrine function of the liver. So when the blood is moving through the sinusoid, remember these hepatocytes, they synthesize plasma proteins. So you have plasma protein being secreted into the blood. So the classification of a liver lobule emphasize the endocrine function of the liver. Because as the blood is moving towards the central vein, these hepatocytes will be draining, right? Secreting into the blood, right? The endocrine function. So this is a good histologic slide. You can actually appreciate this hexagonal shape of the liver lobule, a central vein right here. You can actually see a portal triad here. This being a vein, portal vein obviously, a portal artery here, and a bile duct. And take note of this, a bile duct is going to be lined by cuboidal to columnar epithelia. Cuboidal to columnar epithelia. That's how you differentiate it. A hepatic artery, obviously, definitely you're going to see the smooth muscle in the tunica media. Another diagram right here. This is your central vein. This is another portal triad here. I can see another portal triad here, right? And yeah, another portal triad being formed here. All right. So definitely an irregular large lumen, that's a characteristic of a vein. So this is a vein, a portal vein, and this becomes your artery because I can see smooth muscle right here, right? And the endothelium is a simple squamous epithelium. But as for the bile duct, it is distinct because you can actually see these clear circular nuclei, meaning that there is a simple cuboidal epithelium. You can actually see here simple cuboidal epithelium and a lumen. So that makes a bile duct. And it's going to be embedded in a connective tissue coming from the what? From the Gleason capsule. Remember those septors, right? Blood supply to the liver. Right, you have to understand that the liver is supplied by a portal vein. A portal vein supplies up to 75% of the blood to the liver. But this blood is high in nutrient content and low oxygen. So remember, the liver needs oxygen. So we're going to have a hepatic artery coming from the celiac trunk of the descending of the abdominal aorta. And that celiac trunk will supply oxygen to the heart, to the liver. You understand now? So you're going to have the hepatic portal system, which is the hepatic portal vein. And then from the portal vein, we're going to branch forming the portal venules and distributing venules, inlet venules, And then now we get into the sinusoids. Whenever we get into the sinusoids, where are we going? To the central vein. Central vein. From the central vein, we form the sublobular vein, and lastly, we form the hepatic vein. So we are going to have about 2 or more hepatic vein that will join and drain into the inferior vena cava. You understand? Let's take a look at the artery. So we have the hepatic artery coming from the celiac trunk. This hepatic artery will branch into interlobular arterioles, into inlet arterioles, and then they will drain into the sinusoids. From the sinusoids, we go straight to the central vein. Straight to the central vein. Is it? Yeah. So the hepatocytes at the periphery of the lobule, they are said to be perilobular cells. And take note of this. These, since they are coming from the portal triad, which is a hepatic artery, which is a high content of oxygen, the perilobular cells or the periphery hepatocytes, they are going to be rich in blood, oxygen, nutrients, right? And usually this space or this area is used for protein synthesis. So that's why you have your plasma proteins, your albumin, your clotting proteins are synthesized, and they undergo secretion into the what? Into the blood. Right. Now let's take a look here. We also have cells that are surrounding the central vein. So this is your central vein. So you have cells that are surrounding your central vein. These are central lobular hepatocytes. These have low oxygen content. Low oxygen content, right? They have lower concentration of oxygen, lower concentration of nutrients, and they are involved in detoxification and glycogen metabolism. But as for these ones that are near the portal, these are periportal hepatocytes. They undergo aerobic metabolism and are active in protein synthesis due to high oxygen and nutrient supply. Now, we also say that, remember, in the portal triad, we have what's called a bile duct. So, liver is synthesized bile, right? So, this bile, it flows in the opposite direction with the blood. So, if blood goes towards the central vein, bile goes away from the central vein. So, this is going to be the movement or how the bile is going to flow. It's going to be synthesized in the hepatocytes, secreted in the bile canaliculi. From the bile canaliculi, we get in the bile ductule. From the bile ductule, we get in the bile duct. And then we form a hepatic duct. From the hepatic duct, we join the right and the left hepatic duct, and then we form a common hepatic duct. And it is the common hepatic duct that will join with the cystic duct to form the bile, to form the what? The common bile duct, right? That will later fuse with the main pancreatic bud, entering the duodenum via the major duodenal papilla. So you can actually see this is your bile. So bile will move away from the central, so it will move in this direction. Blood will move in this direction. So they have an opposite. Now let us take a look at what you call a hepatocyte. Where does it originate from? Endoderm. Remember, good. So this is a polyhedral cell with about 6 or more surfaces, highly eosinophilic cytoplasm with many mitochondria, right? Because there's protein synthesis and a lot of things. So It is highly eosinophilic, right? The apical and the basal surfaces are encountered with blood capillaries, and they have microvilli, which we said these microvilli they expand into the space of this, right? Take note of this. The lateral surfaces they are joined by tight junctions, and then they form a system of bile canaliculi where the bile is going to be secreted in. And these bile canaliculi are the ones that will join to form bile ducts, form a bile duct, hepatic duct, and so forth. Cell produce proteins. The hepatocyte, its main function is to secrete proteins. Your clotting proteins like fibrinogen, your plasma protein, your colloid, for example, your albumin, they are all secreted by the bile, by the hepatocytes, synthesized by the hepatocytes. They also undergo detoxification by oxidation, methylation, conjugation, and so forth. And it is a storage unit for lipids, glycogen, and vitamins. Right. Take note of this. A damaged liver can regenerate by mitosis of the hepatocyte to form the lobules. So yes, the liver is able to regenerate. But if there is a repeated insults, right, or repeated injury of the liver, uh, the hepatocytes will enter the quiescent stage, which is the G0 stage. They no longer divide. So therefore, you are going to have the ether cells. Remember the ether cells in the space of this. They take over and they become the myofibrils, right? They will start to secrete and synthesize collagenous fibers, and they will form scar tissue, which will lead to fibrosis of the liver. So this fibrosis is very dangerous because it will be associated with the liver cirrhosis. And liver cirrhosis will lead to pulmonary, portal hypertension. And portal hypertension, remember, it leads to what? Also for Gall, varicocele, if you remember, right? So that's where it starts. So when you see also for varicocele, you also have to predict pulmonary hypertension. And pulmonary hypertension may be caused by liver cirrhosis, which is the increase in the deposition of collagen by those ether cells in the space of this. Now, so this is where you have to get, if you are to be asked about the liver, you have to know what is a liver lobule, what it does, a portal lobule, what it does, and the liver acinus, what it does. These are the main functional unit of the liver divided into these three. Let's start with the classic liver lobe. We have already spoken of it. That's the one that we said it's hexagonal in shape. There's a central vein in the center, and it's going to be surrounded by up to 6 portal canals, right? And we said this emphasizes the endocrine function of the liver. Blood flow towards the central vein, and the hepatocytes will be secreting into the blood. So what's that? That's endocrine function. We have another lobule that is called the portal lobule. The portal lobule. So the portal lobule, it is triangular in shape, and he has a portal space in the center, surrounded by 3 central veins, right? It emphasizes the exocrine function of the liver. So it's sort of like predicts or emphasize the movement of bile in the what? In the liver, parenchyma. Right. So you can actually see this is a central vein, this is another central vein, this is another central vein. So if I join this central vein, da, 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 you're going to form a triangle. That's your portal triangle. lobule. And you're going to have a portal triad at the center. So bile is going to move from this side towards the portal triad, So it shows the exocrine function. As for the hepatic lobule, it shows the endocrine, because you have blood going towards the central vein, right? Towards the central vein, which is an exocrine function. Now, let me talk about the liver, or the hepatic acinus. So the hepatic acinus is diamond shaped. It is made by two central veins and two portal canals. This is the true functional unit of the liver. Take note of this, right? What does it emphasize? It emphasizes blood supply and oxygen, blood supply, oxygen supply, metabolic regulation of the liver. Let's take a look. All right. So we say the two central veins and two portal canals. Right here is the central vein. This is the central vein. This is a portal canal. This is a portal canal. So if I am to draw, it's going to be diamond shaped. Da, 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 da. You understand? So we have three zones. Zone number one. Zone number one. What is happening in zone number one? Take note. Zone number one, it gets the most oxygen and nutrient supply. Why? Because it is closer to the portal triad. So this is where most of oxidative metabolism, such as protein synthesis, take place in zone number one, right? Zone number two is an intermediate range. So it performs intermediate, right, metabolic function, because it is between zone number one and zone number three, right? And this, away from zone number two, we have this space, zone number three, that is now closer to the central vein. So hepatocytes in zone number three gets the least nutrient and oxygen supply. So that's where most of your detoxification occurs, anaerobic metabolism occurs. So this becomes your hepatic acinus, right? So this is a portal, this is a central vein, another central vein here. So this is a portal, sorry, this is a portal vein. You can actually see this is a portal vein, right? So this is a portal triad, because you can see connective tissue around, right? There's no connective tissue surrounding this. This is just a vein surrounded by hepatic cords. So this is a region closer to the portal triad, zone 1, zone 2, zone 3. So you can actually see zone 1 is a bit basophilic. Why? Because there's going to be abundant reticulum, your rough endoplasmic reticulum for protein synthesis, right? And as we move from this, you can actually see we are now getting more eosinophilic and more eosinophilic. At this position, there is not much of protein synthesis that takes place here, right? So you're going to have low concentration of rough endoplasmic reticulum, therefore it's eosinophilic. So we are done with the liver. Let's take a look at the gallbladder. The gallbladder. Good. Right. So what is the function of the gallbladder? The gallbladder does not secrete bile, but it stores and concentrates the bile. NB does not secrete, it concentrates, right? So it is a mucosa It is no submucosa. This has to be corrected. It is no submucosa, right? But it is the um, muscularis mucosa is there, right? And some and serosa. So take that. We have no submucosa. You understand now? So we have the mucosa, absent of submucosa, muscularis, and serosa. They are present. The mucosa is has what you call temporal folds. So if you're gonna look at it in the inside, it's gonna have folds, but these are temporal folds. They can stretch, right? When the bladder is filled, when the gallbladder is filled, right? We also have mucosal uh, glands which are present in the neck region. The lamina propria is rich in elastic fibers and reticular fibers, right? And the muscularis mucosa is absent. The muscularis externa is present. So what is absent? One, the submucosa is absent. The muscularis mucosa is absent, right? Then the submucosa is absent, right? The muscularis is atypical of the GIT, the inner circular and so forth. So you can actually appreciate smooth muscle right here, right? Folding of the mucosa. You can actually see the lamina propria, and you cannot see the submucosa here, right? So you can actually see this columnar, simple columnar epithelia, lamina propria, right here, forming the core of the what? Of the fold. Right, so that's everything about the liver and the gallbladder. Thank you so much. Take care.